You know, it's this near universal human experience. You wake up, you stumble into the bathroom, grab your toothbrush, and, you just start scrubbing.
Right. Autopilot.
Exactly. And while you're doing that, you probably aren't thinking about the fact that you are committing a daily act of, like, urban destruction.
Yeah, you're physically tearing down a microscopic city.
Which is just wild.
It is a highly organized, heavily fortified metropolis that just happens to be built on your molars.
So today we're going on a deep dive into the microscopic world of what microbiologists call biofilms. Looking at the stack of sources for this one, scientific articles, historical research, clinical papers, the mission here is to really understand how these microbial communities stick together.
How they communicate and how they become nearly indestructible. Yeah.
Because if you're like me, you probably grew up picturing bacteria as these lonely, free-floating little cells just aimlessly swimming around under a microscope.
Which is a very common misconception.
Yeah, right. But going through this research, it turns out they are master city planners. Okay, let's unpack this. Looking at the plaque we're brushing away every morning, what is the actual blueprint of a biofilm?
So to understand the blueprint, we really first have to look at the structural foundation. A biofilm is a community of microbial cells. And that's usually bacteria, but fungi and algae do this too, that encase themselves in this self-produced matrix.
Oh.
The scientific term in the literature is extracellular polymeric substances, or EPS.
I have to say, extracellular polymeric substances sounds like something you'd buy at a hardware store to seal a driveway.
I mean, you're not wrong. It functions very much like an industrial sealant, but just vastly more complex.
Right.
When bacteria enter a biofilm, their entire physiology changes. I mean, their gene expression, their growth rate, their stress tolerance, it all fundamentally shifts compared to when they're just, floating around on their own in liquid.
And that floating state, the microbiologists call that planktonic, right?
Exactly, planktonic.
Because I was reading through the historical notes you gathered for this, and it seems like for decades, the scientific community had a massive blind spot here.
Oh, absolutely huge.
They were essentially studying bacteria in captivity, right? Growing these planktonic, free-floating cells in lab broths and just assuming that was their natural behavior.
Yeah, the lab flask was the default for a very long time, mostly just because it was easier to measure and control.
That makes sense.
But that started to fracture. In the 1940s, marine microbiologists like Claude Zobel in 1943 started noticing something weird. If you put seawater in a glass container, the vast majority of the bacteria didn't stay in the water.
They attached to the glass.
Exactly. They rushed to colonize the glass walls. And that was this massive clue that microbes actually preferred to attach to surfaces.
But it took decades for that to actually change how we view microbiology, didn't it?
It did, yeah. The real paradigm shift came much later in the late 1970s, spearheaded by Bill Costerton.
He's the one often referred to as the father of biofilm biology, right?
Right. He and his colleagues, Gysi and Cheng, published a landmark piece in Scientific American in 1978 called How Bacteria Stick.
Great title.
Very to the point. But they argued that the standard way we'd been studying bacteria and liquid broth was, well, an artificial state. In nature, bacteria don't want to float alone. They want to attach to surfaces and build these dense, cooperative communities.
Which implies that for decades, medicine and industry were fighting the wrong version of the bacteria.
Right. If we connect this to the bigger picture, that is the terrifying implication. Doctors and engineers had been developing antibiotics and disinfectants based on how they affected free-floating cells in a test tube.
Oh, wow. So they were testing weapons against, like, an army out in an open field.
Yes. But in a human body or inside a pipeline, the bacteria were hiding inside these fortified cities. The drugs would work in the lab and totally fail in the real world.
That makes total sense for something like plaque on a tooth or, I don't know, a barnacle on a ship. But if they need a solid surface to build on, how do they survive in fluids? Like, how does a biofilm exist in a bloodstream or in lungs?
Well, that was the next major evolution in our understanding. Early on, researchers thought a hard surface was a strict requirement. But pivotal papers, like a major 2004 review in Nature Reviews Microbiology by Hall Studley, Costerton, and Studley, they broadened the definition.
Okay, how so?
They realized these microbial cities don't actually need a solid foundation at all. They can exist as floating aggregates.
Wait, floating cities?
Yeah, we see this in the mucus of a cystic fibrosis lung, or as suspended clumps, what they call flox-thin wastewater treatment tanks. The core behavior, the matrix, the shared gene expression, the defenses, it's all there, even without a rock to cling to.
So it's less like barnacles stuck to a pier and more like a floating raft of fire ants locking their bodies together to survive a flood.
Yeah, that is a remarkably accurate, if slightly terrifying, way to picture it. And that distinction changes how doctors treat infections. They now know to look for biofilm-style defenses, even when an infection is freely floating within the body.
Okay, let's talk about those defenses then, and what is actually holding these floating rafts together. Because when I picture pond scum or dental plaque, the word slime comes to mind. It just seems like passive dirt.
That is probably the biggest myth we need to dismantle right here. Biofilms are not passive slime.
Not at all.
No, that EPS matrix is highly organized armor. It can be over 90% water, but structurally it acts simultaneously as a scaffold, a glue, a chemical filter, and a physical shield.
That's a lot of jobs for one matrix.
It's incredible engineering. It's a cocktail of polysaccharides, proteins, and lipids. But one of the most counterintuitive building blocks they use is extracellular DNA, or eDNA.
Yeah, I struggled to wrap my head around this in the reading. They use their own DNA as rebar for their buildings.
Yeah.
But DNA is the instruction manual for life. You don't just like throw it in the concrete mix.
I know, it sounds totally reckless for an individual cell. In fact, bacteria will actively release this DNA by bursting themselves open, a process called self-alkylasis, just to provide structural integrity to the matrix.
Wait, they use their own dead as mortar.
Essentially, yes. But it highlights something profound about this. A biofilm is about the collective, not the individual. Individual cells will sacrifice themselves to reinforce the walls so the rest of the colony survives.
And a structure that complex doesn't just appear out of nowhere. The research breaks down the construction process into a life cycle with 5 distinct stages.
Right, let's walk through those mechanics. Stage 1 is reversible attachment.
Reversible, okay.
3 floating cells bump into a surface. At this point, they're just loosely held by weak physical forces. They can detach and swim away if they want.
So they're just window shopping for real estate. But what triggers them to actually commit and move to stage 2?
It often comes down to mechanical sensing. When a bacterium's flagellum, its little tail stops spinning because it's pinned against the surface, that mechanical stress triggers a cascade of chemical signals inside the cell.
Oh, wow.
Yeah, and that initiates stage 2, irreversible attachment. The cells permanently anchor down and switch on the specific genes to start pumping out that EPS matrix material.
Then comes stage 3, the microcolony formation. That's where they start dividing and clustering together.
Yes, they multiply and stack on top of each other. But stage 4 maturation is where it gets incredibly complex.
But this is the city building.
Exactly. The colony develops full 3-dimensional architecture.
Yeah.
It's not just a solid lump of cells. They form rudimentary circulatory channels, essentially convection channels driven by fluid dynamics.
Can move things around.
Right, so water and nutrients can flow deep into the structure and waste can be flushed out.
They literally build plumbing.
They do. And finally, you have stage 5, which is dispersion. The matrix enzymes break down in specific spots, and cells detach either individually or in big clumps and swim off to seed new colonies elsewhere.
Which explains why chronic infections are so maddening. You might clear the top layer with a drug, but the colony is actively designed to keep reseeding itself.
Exactly.
And it's wild to think Antony van Leeuwenhoek actually documented this in 1683. He looked at his own dental plaque under a primitive hand-ground lens and saw swarms of what he called animalcules.
Right, he was looking at dispersion happening in real time.
But if you have this massive multi-story structure with water channels and a complex life cycle, how do single-celled organisms organize all of this? I mean, they don't have a central nervous system.
No, they don't have a brain, but they possess a highly sophisticated chemical communication network called quorum sensing.
Quorum sensing.
All right, bacteria constantly release small signaling molecules into their environment. In gram-negative bacteria, these are commonly molecules called AHLs. As the population of bacteria grows, the concentration of these molecules builds up in the surrounding fluid.
So it's basically a chemical head count.
That's a great way to put it. The crucial mechanism is that the bacteria have receptors for these molecules. But the receptors only trigger a change in the cell's behavior when the chemical concentration crosses a specific threshold.
A tipping point.
Exactly. Once it hits that threshold, it signals to every cell simultaneously. Enough of us are here to make an impact. And only then do they collectively switch on group behaviors, like producing the EPS matrix or releasing virulence factors.
That threshold concept really explains the bioluminescent squid example I saw in the sources.
Oh, that's a classic one.
Right. So the Hawaiian bobtail squid has this symbiotic relationship with the bacteria called Vibrio fischeri. When those bacteria are floating alone in the ocean, they're dark. But when they pack tightly into the squid's light organ, the quorum-sensing threshold is breached and they all light up at the exact same time.
Providing camouflage for the squid.
Yeah, it's amazing.
And foundational work by researchers like Bonnie Bassler and Kenneth Nielsen really mapped that out. It's a perfect illustration of how chemical signaling allows for coordinated, energy-intensive group strategy, but only when there is safety in numbers.
Okay, here's where it gets really interesting, though. The sources highlight a study from 2015 led by Gurl Sewell on Bacillus subtilis biofilms. It showed they don't just talk with chemicals.
No, they don't.
They communicate electrically. How does a bacterium send an electrical signal without a nerve cell?
It's just phenomenal mechanism design. They use potassium ion channels.
Yeah.
When cells deep inside the biofilm start running out of nutrients, they open these channels, releasing positively charged potassium ions. This triggers neighboring cells to do the same, creating a wave of electrical depolarization that sweeps across the entire colony, all the way to the outer edge.
So the starving cells in the center are literally shocking the cells on the outside.
But in a way, yeah, that electrical wave forces the outer cells to temporarily pause their own growth.
Oh, I see.
Right, by stopping the outer cells from consuming all the nutrients, the food can finally diffuse deep into the center to feed the starving interior. They're synchronizing their metabolic activity between distant cells in a way that is eerily similar to how human neurons signal in a brain.
If they have chemical voting and electrical resource management, it sounds like a perfect utopian society. But biologically, where there is a public resource, there's usually something trying to exploit it.
Yes, and this raises an important question, which is actually a major debate among evolutionary biologists like Stuart West.
What's the debate?
Is quorum sensing genuinely cooperative communication, or is it a public good that gets exploited? Because producing these chemical signals and building that EPS armor, it costs a massive amount of metabolic energy.
Right, it's expensive for the cell.
Exactly. So inevitably, mutations arise, creating what we call cheater cells.
Microbes that just stop producing the matrix.
Yes. They stop making the glue or the signal. They just hang out inside the city, reaping all the benefits of the physical protection and the plumbing without paying the metabolic tax of contributing to it.
But if they're draining resources without contributing, why doesn't the whole biofilm just collapse under the weight of all these cheaters?
That is the active evolutionary battleground. Biofilms employ policing mechanisms, often restricting the benefits of the matrix to close kin, or linking the production of the public good to private genes essential for the cheater's own survival.
Oh, clever.
It's a constant arms race just to maintain the structural integrity of the fortress.
And clearly the fortress holds, because these cities are notoriously difficult to destroy. Which brings us to how they survive our most potent weapons, like antibiotics. There seems to be a major public misconception here.
There is a very big one.
Because when we hear about bacteria surviving antibiotics, the immediate thought is genetic superbugs, bacteria that have mutated their DNA to become permanently immune to a drug.
Right, that is antibiotic resistance. And while resistance certainly can happen in a biofilm, the primary survival tactic of these communities is entirely different.
It's not mutation.
No, it's about antibiotic tolerance or persistence. The difference is fundamental to how we treat them.
Walk us through the mechanics of tolerance then, because the sources place heavy emphasis on this distinction.
Okay, think about the environment deep inside one of these mature biofilms. The outer layers of active, multiplying cells are rapidly consuming all the oxygen and the nutrients.
Because they're on the front lines.
Right, so the cells trapped in the deep interior are suffocating and starving. In response, they undergo a physiological shift. They enter a dormant, radically slow metabolizing state. They become what we call persister cells.
And why does going dormant protect them from a drug like penicillin?
Because of how the drug actually works. Most of our best antibiotics function by disrupting active biological processes.
Right.
Penicillin, for example, attacks the enzymes that build the bacterial cell wall.
Right.
But if a persister cell is dormant, it isn't building a cell wall.
So it's like a burglar alarm that only detects movement. If the bacteria just stand perfectly still by going dormant, the antibiotic has no active process to attack.
Exactly. The drug just washes over them and they sleep right through it.
Wow.
And this isn't a modern phenomenon either. Joseph Bigger documented this in 1944. He exposed a population of Staphylococcus to penicillin in a lab setting, and a tiny fraction survived.
The persister cells.
Right. But the kicker was, when he took those surviving persister cells out of the biofilm environment, gave them fresh nutrients so they woke up, and exposed them to penicillin again, they died instantly.
Because they weren't genetically resistant.
Exactly. They hadn't mutated, they just temporarily changed their state to survive the siege.
But the physical matrix itself has to play a role in stopping the drugs too, right? It's not just the cells hiding in the basement.
Oh, absolutely. Mathematical modeling, like the work done by Philip Stewart, shows how the EPS matrix acts as a chemical sponge.
A sponge.
Yeah. Many antibiotics are positively charged molecules. The EPS matrix is heavily laced with negatively charged DNA and proteins. So as the drug tries to diffuse into the biofilm, it literally binds to the matrix and gets neutralized before it ever reaches the deeper cells.
It gets trapped in the walls.
Right. Today, the consensus is that biofilm survival is a brutal combination of both. The matrix chemically slowing the drug down, and the persister cells sleeping out whatever makes it through.
It really highlights how a biofilm acts almost like a single multicellular organism. You've got the fast-growing active workers on the outside taking the hit, and the dormant survival bunker cells on the inside preserving the lineage.
Yeah, researchers like Coulter and Watnick popularized the term city of microbes to describe this. They point to this as a form of primitive multicellularity. They're actively dividing labor for the survival of the whole.
And when that division of labor happens inside a human being, the stakes are incredibly high. The medical implications here are just staggering.
They really are. One of the most studied and consequential examples is the bacterium Pseudomonas aeruginosa in patients with cystic fibrosis.
Cystic fibrosis, right?
The mucus in a CF lung provides an ideal stagnant environment. When Pseudomonas colonizes it, the bacteria undergo a phase variation into a mucoid phenotype. They drastically overproduce a sticky polysaccharide called alginate.
What does that actually do to the lung environment, though?
It thickens the biofilm matrix to an extreme degree. It makes it so viscous that the patient's immune cells, macrophages, and neutrophils physically cannot penetrate the slime to engulf the bacteria.
Oh, it sounds awful.
It is. The immune system keeps firing inflammatory chemicals at the biofilm, which ends up damaging the patient's own lung tissue without clearing the infection.
And this extends far beyond cystic fibrosis. I saw Costerton's 1999 science paper estimating that biofilms are implicated in the vast majority of persistent bacterial infections in humans.
It's a widely cited estimate, yes.
We're talking about bacteria colonizing medical implants like catheters, prosthetic joints, or heart valves, where the immune system has an even harder time reaching them.
And it doesn't stop at the hospital doors. The mechanics of the EPS matrix create absolute nightmares for industrial infrastructure, too.
Yeah, the sources mention the food safety industry battling Listeria monocytogenes. It hides out in biofilms on stainless steel processing equipment, where the EPS matrix completely shields the cells from standard sanitation chemicals. Or Legionella pneumophila, which causes Legionnaire's disease. It survives inside biofilms in cooling towers because the matrix chemically neutralizes the chlorine treatments before they can penetrate to the Legionella cells.
Ship hulls, oil pipelines, municipal water systems. The global cost of dealing with biofouling and corrosion caused by these microbial cities is just astronomical.
Given how stubborn and dangerous they are, it's not surprising that the consumer wellness industry has totally latched onto the concept. You go online now and you see supplements marketed as biofilm disruptors, claiming to cure chronic gut issues or brain fog by breaking down microbial walls.
Yeah, it's a classic case of marketing sprinting ahead of clinical evidence.
Right.
To be fair, the core biology we've discussed, the matrix, the persister cells, the quorum sensing, is entirely legitimate in clinical settings. But the leap to taking an unregulated over-the-counter enzyme and assuming it will safely and specifically dissolve a pathogenic biofilm in your gut without harming anything else, well, the rigorous evidence for that is often incredibly thin.
So what does this all mean? Because not all biofilms are villains, right? We actually need them.
Desperately. Most biofilms in nature are essential. I mean, they are the literal workhorses of our wastewater treatment plants. Engineers purposefully cultivate massive biofilm structures on trickling filters or in activated sludge so the microbes will consume and break down human waste before the water is released.
Oh, nice.
They cycle nutrients in agricultural soil. And inside a healthy human gut, beneficial mucosal biofilms act as a protective barrier, physically blocking harmful invaders from reaching our tissue.
So looking at the big picture of what we've pulled from these sources today, we're really dealing with a fundamental shift in how we understand microscopic life. And we have 5 big takeaways for you. Number 1, a biofilm isn't just bacteria stuck together. It's an engineered structure built with a self-manufactured armor of sugars, proteins, and even their own DNA.
Number 2, their ability to survive our medical interventions relies heavily on physiological tolerance. It's the dormant persister cells surviving antibiotics by simply halting their active processes, protected by a matrix that acts as a chemical sponge.
Right. Number 3, it's all coordinated by quorum sensing and electrical signaling, allowing them to count themselves and trigger group strategies only when they have safety in numbers.
Number 4, Constantine's historical reframing was vital. The free-floating, planktonic bacteria we studied in lab flasks for decades was an artificial state. Structured, cooperative biofilms are the true default in nature.
And number 5, biofilms aren't inherently evil. They are structural marvels that can cause devastating chronic infections, sure, but they also clean our water and protect our digestive system.
Exactly.
Which brings me to one final thought from the research that I haven't been able to stop thinking about. We talked about how advanced the city building is. Well, microbial mats, which are essentially ancient biofilms, have been found preserved as fossilized structures called stromatolites. And those fossils date back roughly 3.5 billion years.
Placing them among the oldest evidence of life on Earth.
So here is something to mull over while you're brushing your teeth tomorrow morning. If microbes figured out division of labor, protective skin, chemical communication, and electrical nervous systems billions of years ago, are our own human bodies, our complex organs and tissues, just a biological knockoff of a blueprint that bacteria perfected first? Thank you for joining us on this deep dive into the source material. We'll see you next time.